Yeah, but don't feel shy about saying anything controversial or anything. This is an open space.
It will just be permanent on the internet, so I think you should just let your hair down.
Don't worry, it's not gonna go on YouTube. Just gonna send it to the people who missed. Out of curiosity, does anyone here, because today's the science section, does anyone here have like a biology background or engineering background, or chemistry or anything? Very cool, awesome. Well, I hope my presentation is digestible for everyone, but feel free to stop me with any questions. I'll wait a few more minutes. I don't know. I think cohort 2, it looked like there were lots of people on the Slack channel, at least.
Yeah, I see like 18 people, but I'm not sure how many of those are like Yumi and Eric and Ashley. So
Yeah.
I hope you'll get a chance to talk about TMAO and whether in the cultured meat that is replicated, or if we can somehow eliminate that so that it reduces the health problems. It's a lot of the reading has been about cholesterol and saturated fats and that sort of thing, but TMAO is a huge issue for heart health and other issues.
So I have not brought up TMAO, although I'm just Googling it now.
And I
yeah,
could,
I've never,
yeah,
I don't even know what that stands for. I never really view this I mean, I'm nascent to this field, but I haven't thought so much about like health implications as much, but I would love to hear opinions on that.
Yeah, I'm less of a nutrition expert,
Mhm.
more so just molecular biology and whatnot. But I'd love to hear your thoughts on it, especially in the discussion section.
Yeah, I think we do
yeah.
cover some like questions about like how we'll be able to like convince people to switch their diet to this, and I think that would fit really well. And whether or not this is natural, so I think that will fit nicely there.
Great. I'll post some information about that too. It's trimethyl something something, but at this hour in the morning, I don't remember the details, but I can put
Okay.
together a little packet because it would help with a lot of people.
Yeah, thanks. All right. So I guess really quickly, could we just go around and I just want to hear like a brief introduction from everyone. I guess we can start with Juan Lucas.
Hi, yeah. So my name is Juan Lucas. I'm actually 17. I'm still in high school, but I'm really interested in cellular agriculture. So I'm just here to learn more and to meet other people who are interested in this field. So, oh yeah, I live in Hong Kong right now.
It's awesome. And next we can go with I'm having a hard time saying names. I guess we can go with Linda.
Hey, I'm Linda Watson. I live in Raleigh, North Carolina, US. I'm a food writer and food activist. So my background is in nutrition and animal rights. I'm the author of Wildly Affordable Organic and run the website and project Cook for Good. So you can find us at cookforgood.com.
Can't wait to check that out. I'm a big fan of cooking myself.
I've
So
actually seen the website.
what was that?
I've seen Linda's website. I've looked through it.
Awesome. Whitney?
Yeah, hi. My name's Whitney, and I just graduated from Emory University. I'm currently working in a biotech company called CellX, and we're actually working on cell culture meat right now. And I'm actually in this project to see more of a customer feedback of how people or the general public is seeing this cell culture industry. And shout out to whoever that's in Hong Kong right now, because I'm in quarantine in Hong Kong right now. Yep.
Cool. 2 people from Hong Kong now. That's awesome. Nadia?
Hi, I'm Nadia. I'm a first year at Brown. I'm interested in, I like to cook, interested in biology and nutrition. So I want to learn more about going plant-based and convincing other people to go plant-based too. And I'm in Singapore right now. So like happy we have a huge Asian crowd.
That is awesome. Dinesh?
Hi, I'm Danish. I'm from southern part of India. I recently graduated with Masters in Biotechnology and Biochemical Engineering.
Cool. Okay. So yeah, some of you, especially maybe Whitney and Dinesh, might have a little bit more background on the science aspect. So feel free, I'm really looking forward to the discussion. Molly will lead. And just before I get started, I will introduce myself, and Molly can introduce herself. So my name's Lauren. I am a 5th year PhD student, about to be a 6th year, so thinking about graduating soon, at Johns Hopkins in the School of Medicine. And the program that I'm in is Program for Molecular Biophysics. And my undergraduate degree is from the University of Cincinnati in Ohio, in biomedical engineering. And this isn't my thesis work necessarily, but I've done some teaching on this topic at Hopkins already. And it's just something I've gotten really passionate about, and I probably will dedicate my career to it, maybe at a biotech company or something. So yeah, feel free to jump in at any time. Molly?
Hi guys, I'm Molly. I'm also a 5th year PhD student in a different program. I'm a cell biologist, but I work on the same floor as Lauren. So we're both at Johns Hopkins in Baltimore. And I did my undergraduate degree in research in Florida State University in Tallahassee. I'm originally from Miami area. And my research also has nothing to do with this, but I think I've talked a lot with like Lauren about this, and her enthusiasm is really like infectious. So I've just joined like the Alternative Protein Project and been part of like developing our education side of that. So like doing journal clubs and working on classes like these. So I'm learning with some of you guys here. Like I said, I'm new to this field.
All right, so we can go ahead and get started. Okay, can everyone see my screen? I'll go into presentation mode.
Yep, we can see it.
All right, so thanks again for coming. Today we're going to be focusing on some of the science behind the 3 pillars that you guys may or may not have learned about from Module 1. Just a reminder, their alternative protein can encompass plant-based, fermentation-derived, or cultured animal products. Essentially, alternative protein is protein that does not come from animals. And so while there are 3 pillars right now in the alternative protein tech, I want to emphasize that some of these can be hybrids of each other. A lot of products use combinations of these. For example, Impossible Foods uses plant-based proteins for their actual burger, but then they use fermentation technology to produce the leg hemoglobin, which provides that irony, meaty, bloody taste. And also, I think in Singapore right now, Just Eats put out their first ever cultured chicken product, which is a combination of plant-based protein and animal cell culture. You also have fermentation is helping to provide materials and growth factors for animal cell culture. And basically, the 3 pillars are can be, very hybrid. And so I'll first talk about some of the science behind plant-based meats. So plant-based meats have been around for a long time. Veggie burgers. I don't know if anyone here has always had veggie burgers. I've been vegetarian since I was 11 years old, and vegan for, on and off the last few years. And so plant-based meats have come a long way, and they are usually made from proteins such as soy or mung bean, pea protein, or other legumes. And the way that you can get this textured, meaty structure is through texturizing processes such as extrusion. And so I'll go into a little bit about the texturizing processes later. So basically, you can get these protein powders from the plant substances, and to get them into a Impossible Burger or Beyond Burger with that texture, that gelatinous texturized material. There is a lot that happens in between here. So you can't just add this flour from the plants directly and use that. There is a lot of processing. So the plant-based meat workflow kind of works like this. So first you're going to source your material. It could be mushrooms or wheat or lentils, whatever. You want to find the optimal protein for your product. I'm sure companies usually try a few different ones before landing on their final choice. Usually there is these plants can be optimized via breeding or genetic engineering. And so this could be just typical GMOs to give it insect resistance or herbicide resistance. However, that's not the only kind of GMOs that occur. Sometimes you might have a taller plant or a thicker plant, more efficient plant, something that will ultimately give you the most efficient product. And so after you harvest your plant proteins, the raw materials, the protein has to be isolated and also functionalized by various mechanical and chemical processes. For example, you'll need to remove a lot of carbohydrates from the plant protein as well as fats, so that you can have the ultimate texture and meat fibrosity. Finally, you can add various ingredients or post processes to create the desired taste and texture, smell, structure. That also includes color and appearance as well. So I'll go into a little more detail. So of course, there's plant protein is first separated from the fats and carbohydrates and also the fiber that's in the initial harvest. And there's several different levels of these plant protein flours. So, well, you have an actual flour, which is 20% protein. That's just if you grind up the bean or grind up the kernel. So these are used in pastas and savory snacks. Protein concentrates have a little bit more chemical processing to enrich for the protein. And that can end up in 55% protein. So these protein concentrates, they can be used in baked goods and bars. Also, some companies will use protein concentrates. They find 55% protein is enough to then use in a texturizing process for a burger. Finally, protein isolates, these require the most physical and chemical processes in order to remove the carbohydrates and lipids and enrich for the protein. It has 80% protein. And So these are usually found in nutrition bars, drink mixes, and meat and dairy alternatives. So oftentimes protein isolates are what are used in the main ingredient for, I think, the Beyond Burger or Impossible Burger. But I do want to emphasize that, these processes can be quite energy intensive, and they can use various chemical processes in order to separate the carbohydrates and the lipids based on their physical chemical properties. And here is one of the most popular methods for texturizing protein, and it's called extrusion. So the extrusion process essentially involves adding soy protein concentrate or isolate, and maybe another protein source such as wheat gluten, whatever your ingredients are. You feed them into this rotating extruder. It's like a screw that's constantly rotating, and it has high pressure, a little bit of moisture, and high temperatures. And so what the high pressure and the high temperature will do is break apart any protein interactions that are already there. And because this is going along the products are going along a laminar flow, the protein interactions will form new interactions with neighboring proteins, and it'll essentially form long fibers. And finally, you'll have a cooling die at the end, which quickly cools the proteins so that they can kind of solidify their new fibrous interactions. And when I say fibrous, this is kind of what I mean. This, was soy protein concentrate or wheat gluten, but now it is forming these long strands. And if you look at a microscope, it kind of does look like skeletal muscle under a microscope. But it's just plant proteins that have reformed their interactions. Now, that's quite an energy intensive process. And so there is a lot of work going on to using lower temperatures or using less power or less pressure. There's another technology that is becoming more popular now called the shear cell. It's pretty similar to extrusion, except it utilizes shear stress, which is essentially if you put opposing forces on an object, you bend it a little bit, and that's shear stress. And so your product will go in between these two cones, and the cones will then rotate, and it can spit out, meaty products that look like this, or something like this. And then of course you can cut them, post-process them, shred them, whatever your final product is supposed to be. And here's a good close-up of a meaty texture produced by shear cell. This, you can see there's some longer fibers with some mini fibers in between. So this just goes to show how powerful these protein interactions are, or how important they are for forming the meaty structure. And then, of course, afterwards, after you cut or shred or grind up the texturized meat afterwards, you can add various things such as flavorings, iron or iron-containing materials to add the meaty taste. Sulfur-containing compounds often provide the roast aroma. And color is an extremely important aspect of meat. You don't want to eat a burger that's gray, for example. So oftentimes, to make it look more bloody, you can add natural colorants such as beet powder, paprika, turmeric, and pH adjusters will also be important there to maintain the color over time, such as citric acid. You also might add binding agents. This meat is a very gelatinous food, And so for a gelatinous texture, various binding agents can be added, such as wheat gluten, xanthan gum, or pectin, and this will hold various meat shreds together. Also, sometimes people use egg as the binding agent, although that's becoming less popular over time since the vegan movement is becoming more popularized. And then some of you might be thinking, okay, what about like a big juicy steak? Like a lot of the processes that I've shown thus far are very useful for making ground beef patties or burgers and stuff that's kind of shredded. But some people really like the big structured products like steak. And so some companies have started utilizing 3D printing in order to offer structured shapes with different fat, blood, and muscle lines that can be plant-based. Now, I want to also emphasize that 3D printing can also be used for cultured meat. And there's lots of companies who are using that as well. Okay, so that was a really quick recap of plant-based meat, but if anyone has any quick questions before I go on to fermentation, just let me know. Okay, I'm not seeing anything, so I will move on. All right, so one of the second pillars is fermentation, and fermentation can mean a lot of different things to various people, depending if you ask someone who's a biochemist or someone who makes wine or someone who studies yeast. But the way that the alternative protein industry describes fermentation is just any growth of microorganisms such as yeast, bacteria, mold, algae to produce ingredients in food. And fermentation can include cultivation of any of these microbial species that I just mentioned. And a lot of these can be grown in bioreactors, which is also the same for cultured meat. But there'll be a slightly different bioreactor if you're growing mold versus some stem cells. So some of these organisms actually might grow outside. You might have seen algae ponds. If you've ever been driving long periods of time down highways, you might see like big algae ponds. So that's not a bioreactor, that's an outdoor algae field. Or some sorts of molds prefer to grow in air, for example. But a lot of the times you're going to be using big bioreactors and just feeding the nutrients into the bioreactor without sunlight or without oxygen. And some of the benefits of fermentation for food production are that microorganisms can grow very rapidly. Lots of these microorganisms have division rates on the order of minutes to hours, whereas mammalian cells and a lot of vertebrate cell lines have division rates on the order of days. So you can have a lot more product in a lot less time. And the microorganisms are usually a lot easier to genetically modify than plants and mammalian cells. So they'll take up these genes. This is a plasmid with various foreign genes, and you can add them into the bacteria cells or the yeast cells very easily. Sometimes as easy as just heating them up in a specific buffer, and then they can grow from a single cell to a big colony just overnight. And because of this, they can readily express any recombinant DNA, meaning like DNA that uses for other genes from other organisms in order to produce the optimal phenotype. And microorganisms, because they're much simpler, they are more efficient at converting calories into protein, which is great from an engineering perspective. And some microorganisms, such as algae, actually, rather than adding CO2 into the atmosphere, they remove it from the atmosphere. And finally, one of the most logistical benefits is that the regulatory pathway for fermentation kind of already exists, because we've already been using bacteria and yeast and algae in food for centuries. You know, if you ever had cheese or wine, you know that those are produced via fermentation. Yeah, so the fermentation workflow is similar to the plant-based one in terms you have to find your best source for whatever you're trying to do, optimize them maybe via cell engineering or finding the optimal culture conditions. And finally, you'll add the cells into a bioreactor with different cell media, culture media. And this bioreactor might be temperature controlled, humidity controlled. And then finally, you can harvest your product and get whether you might be just using the entire organism, or you might be purifying something specific from it. So there's a few different kinds of fermentation. And fermentation, as I mentioned before, it does enable other sectors within the alternative protein industry. So fermentation might allow some plant-based meats to be a little bit better with, for example, the addition of like hemoglobin. Or you might be able to use fermentation to have microorganisms grow growth factors that might help the cultured meat industry. Or you might take the entire organism and eat it as a standalone protein source. So traditional fermentation has been used in the food industry for centuries, as I mentioned. All of these products use microorganisms in some way. This is tempeh, alcohol, cheese, bread, kimchi, sauerkraut, and many other things. So traditionally, for the traditional fermentation usually uses these two pathways, where when you don't have, when you're in an anaerobic environment, meaning without oxygen, the yeast in the presence of glucose will shift from a respiration pathway to a fermentation pathway, which produces ethanol as a byproduct. And bacteria fermentation, because they have different enzymes within them, they will produce, for example, lactic acid, acetic acid, formic acid, which kind of produces that nice tangy flavor, or the sharp flavor in cheeses. And now biomass fermentation is also becoming more popular now. That's when you take the entire cell of the microorganisms, and those produce the meaty texture. So I'll actually talk about corn in a little bit. So corn is, they use a filamentous fungus, which looks like this, and they grow it in a bioreactor continuously. And at the end, they will heat the filamentous fungus to a point where it will die. And then they will collect all the fungus. They'll use some binding agents to combine it, and ultimately use the fungus, which is really high in a protein called mycoprotein, to create various products like steaks or chickens or ground meatless grounds in this case. And you're eating the entire organism, but it has been killed. So this is technically a mold. Some people are weird about it. They're like, oh, you're eating mold, but it is killed. And it's very high in protein and very nutritious. Now, precision fermentation is another one that has recently taken off in the fermentation field. So there's a few of these startups that just came in the last several years. Basically, instead of basically you are purifying very specific components from the microorganisms. And I'll give an example. So Perfect Day uses fungus to produce casein and whey proteins just by using the casein and whey DNA from the cow, which they didn't have to take the DNA from the cow directly. They could actually just synthesize the DNA with the known sequence, because most animals, especially livestock, have been fully sequenced with their DNA. So I'll share this little commercial for Perfect Day. Yeah, I always really like that cow in the on the left hand corner says on vacation. Makes me very happy because when you have the DNA sequences of all these animals, you don't need to go back to them. You can just synthesize the DNA and produce it in large scale in a microorganism. And Perfect Day just happens to use fungus. Oops. So recombinant DNA essentially involves taking a gene from one organism and putting it into another. So the two examples that I've talked about in this presentation are casein DNA from cows can be cloned into a DNA plasmid that is ultimately expressed in fungus. So this little gene fragment might be put into DNA that can be then added to the organism. And the other example is leg hemoglobin DNA from legumes can be cloned into a DNA plasmid expressed in yeast. Okay, finally, I will just quickly go through some cultivated meat principles. I mean, you could have entire courses on this, but I'll just cover some of the basics. Okay, so cultivated meat is the in vitro culture of animal stem cells to grow muscle, fat, or vascular tissue as a meat product. And this is, to me, this has been kind of the most exciting developments, and it's kind of happened right after the big boom in tissue engineering, which started in the 90s. And so cultivated meat has borrowed a lot from tissue engineering. So what is meat? To make cultured meat and to make it good, you have to understand what are all the components in meat that give it its unique taste and texture. It's 80 to 90% muscle fibers, 5 to 10% connective tissue, 5 to 10% fat, and 0.3% blood. So cultivated meat, this is just a big synopsis. Essentially, you'll first take a biopsy, from an animal. And so this can be done under anesthesia, or you could collect it as a byproduct at a slaughterhouse. But I like to think that hopefully they're getting all of their, I would like to think of a living cow as the donor of this biopsy. And then you can isolate the cells from your biopsy and basically isolate the stem cells. And so the stem cells can become any sort of cell that they want. So they could become a blood cell, they could become a muscle cell, or they can become a fat cell, or fibroblasts to have connective tissue. So that all depends on the environment around them. But first you will want to expand the cells that you've isolated, the stem cells, and then you can downstream that you can mature them or differentiate them so that they can become whatever they're destined to become. And that the differentiation process, whether they become a muscle cell or a fat cell, that depends on the scaffold that they're on and a lot of other conditions, whether there's like movement in the bioreactor, and also differentiation factors, which will be found in the media. After the tissues have matured and they've grown, they can be collected and then processed. This kind of looks a little gross, I think, but they can be processed into your meat product. And so this cultivated meat workflow is essentially just a summary of the previous slide. But yeah, I just want to emphasize that you can based on the culture conditions, you can differentiate into fat, muscle, or fibroblast cells, or other cells. But these are the ones relevant to meat. And then grow them up. So the scaffolds, that is essentially what the cell is growing on. They are very important considerations for growing meat in 3D. Because cells don't like to just grow into the air or into the media, they like to form connections. And so the scaffold research is actually very, very big right now. In fact, a lot of research is going into finding food grade edible scaffolding networks that can just be part of the food or removed and not toxic. So for example, here's a decellularized spinach leaf, which you can see it's just the vasculature of the cell, the extracellular matrix. And this actually was found in some studies to be a good scaffold for certain kinds of meat cells. But there's a lot of decellularized scaffold research going on right now. And if you want to scale this up, you need it to be automated and you need to be a very efficient process. So cultured meat bioreactors will be probably a little bit more involved than fermentation bioreactors. You can inoculate your seed strain, basically just a few cells that you want to grow up, so you can proliferate them in this cell proliferation bioreactor. And then if you wanted this to be an automated process, you'll want, after the cells have proliferated, you'll want them to move directly to a tissue perfusion bioreactor where they might attach to some scaffolds, some specific scaffolds, depending on what you're differentiating them into. And they can grow in larger amounts and differentiate here right after, just directly from the proliferation bioreactor. And finally, I want to talk about the cell culture media. Culture media, it contains everything. It's like the juice that all the cells need. So of course they need glucose, amino acids, vitamins, inorganic salts. If you're differentiating them, they need their differentiation factors. And then there's also serum. Serum is often taken from dead fetal cows, which is extremely gross. But the serum actually has a lot of the growth factors and hormones. And I put this question mark here because we actually don't even know like the extent of everything that's in the serum that causes cells in a tissue culture environment to grow. But the serum is actually the most expensive part, and differentiation factors. So cell culture media is probably the major pain point of cultivated meat technology now. And there's a lot of research going into media optimization. 99% of cultivated meat cost is from media and its growth factors right now. But cultivated meat was $1.3 million per pound in 2013. That's when the first cultured meat burger was made, but it is about $50 per pound now. So we have made a lot of progress, and I believe we will continue to make progress, and other sectors such as fermentation will help with that. So that concludes the lecture. Sorry, I was trying to get basically a whole semester's worth of content into 30 minutes, or I guess it ended up being like 35. But I will leave it to Molly to start the discussion now, and we really want to hear from you guys.
Yeah, so just really quickly, does anyone have any questions on the background slides before we get into some of the conceptual questions? And our like inboxes are open. We're gonna put our like emails on the last slide, but if not, then we can just start with the discussion questions, and we're really happy to hear from you guys. And so the first, oh, go ahead.
Sorry, I just have a question about categorization, about how would you differentiate cell expansion with differentiation? Or like, would you put scaffolding under cell expansion or in differentiation or maturation?
I think scaffolds are necessary at like every step, but I mean, definitely the differentiation process. So and I also want to admit, I have not done this myself. I've just done a lot of reading on it. But I think it is important in the maturation process, but also the differentiation process. Is that your question?
I suppose so. Like, similar with you, I've read so many different ways of describing this process, but then there's like, so where does scaffold and bioreactors comes in the process? Because or you have the maturation, but then to my understanding, scaffolding or bioreactors and also the cell culture media are important throughout this whole process, but then people tend to talk about those much later on. So I'm just a bit, if there's a uniform concept of where these things should be put, I guess that's my question.
Yeah, I also think it probably depends on the cell. Like, so in the cell proliferate, generally cells do not like to grow on just in suspension. I mean, you have some cells that are designed for suspension, but I think stem cells often like to grow on something. And then for the scaffolding network, I mean, I know that the scaffold, if you want to grow like into a 3D environment, I think that's where the scaffold will be extremely important so that it can actually move up, because a lot of these cells like to prefer to grow in a monolayer, and because they have something to attach to. But then if you have a scaffold that moves in 3D, then it can move up, essentially. And so, I think the maturation and the differentiation process kind of go hand in hand in that way. But like all the tissues need to mature, but how they mature and what they differentiate into depends on what they're actually on. So I don't know if your question is the scaffold really important in the proliferation process? I would imagine it probably depends on the cells.
I think I also with the disclaimer that I've never done it either, but I was attending a few seminars about this technology, and I think the cell proliferation, to my understanding, looked like it was somewhat independent of the scaffold, and the reason they were saying that was because there's an issue with the scaffold that when things are growing in 3D and they're very dense in the middle, there's a problem with getting oxygen and like nutrients to that area. So I think as I mentioned, to my understanding, it's best to like bulk up the amount of cells like independent of the scaffold and then put them onto the scaffold so that they can like seed themselves in like an orientation that's conducive to like the flow of media, as opposed to rapidly dividing on that structure and having less control of how much you can distribute and I think it's called perfusion, like perfuse the media to the inside of the scaffold. At least for the talk that I was listening to, that's how I understood it. And that was more at like the bench scale and not necessarily the industry scale. And I think the differentiation in that case occurred like prior to the both the increasing the proliferation and the scaffolding.
Oh, interesting. Yeah, I think you can, I'm sure many companies right now are probably doing it in different ways, but
Yeah, I think so.
Yeah, that makes so much sense.
Okay,
Thank
any
you.
other questions? Okay, then let's get to the first question, which I think the first one we had about like we want to see we mentioned in the beginning of the talk that these 3 pillars of research, like the plant-based fermentation and cellular agriculture, are not independent. And we did tend to present them like independently, like throughout the talk. So we wanted to see how you guys envision like each of these different pillars helping other pillars. Like for example, Lauren mentioned that the plant-based burger for like Impossible Meat, they use like precision fermentation to get the hemoglobin molecule. But we wanted to ask from a different angle, like how do you think that like plant and fermentation products can help cellular agriculture? And if anyone has any ideas related to this.
I guess like from a different perspective, not thinking just like technological, but maybe they could work together in promoting the need for like alternative protein in general as a whole, and like getting support from both the public as well as the government in terms of policy.
Yeah, so like I think that that's a really good idea. Like if we focus, I think we will be talking about like the kind of public like their influence and how they perceive this technology. So if we focus, like I think they'll be in general more willing to accept like the plant-based and fermentation in the beginning. So if we like work to like really increase our technology there, then they might be more like willing to accept the like as the way that I perceive it, like the more extreme version, which is like the lab grown meat. And like one example to answer this question as well is like exactly the idea that like that Lauren mentioned for Impossible Foods, where you use precision fermentation to improve plant-based. You can also use this type of fermentation to create growth factors. Like you can ask your bacteria and your yeast to create the growth factors that you need to grow the cells in the tissue culture media. And that would be a huge advancement, because it would like bypass the fetal bovine serum need, which is that's what she was mentioning, where you have to take the blood from many like infant cows or fetal cows. Any other ideas for this one? I think one more that I heard recently, I went to a talk, and this one kind of opened my mind a little bit, that there's a lot of like, when we process crops, there's a lot of byproduct. Like there's these crops that are called like pulse crops, like they're like chickpeas and legumes and beans, and their waste product or something that is like not really, it's like a tough structure that is not good for like I guess like human consumption directly. But somebody in this talk mentioned that we can utilize those byproducts as the actual scaffold for growing cellular agriculture and like those types of muscle cells. And that would be awesome, because it's like recycling and reducing your waste. And it's similar to what Lauren brought up with like the decellularized spinach. Like it's just basically plant byproduct, and we know that it's biodegradable and edible. So it would be a great use to combine, like, for example, plant-based product with cellular agriculture. Okay, we can go to the next question. And so that first question, I don't know if you guys got the list of readings, but the first question was from an article written by she's a PhD who works at the Good Food Institute. Her name is Dr. Liz Speck, and that was like a short like magazine article. So we highly recommend reading that one. It was really digestible to like also non-science audience. And the second question we have is based off of fermentation. And does anyone have any idea of like weaknesses or drawbacks that come from like fermentation or even plant-based technologies in general? Like this can be like at the level of the science or like the consumer demand for it.
Well, I think I read somewhere that there is like a similar level of protein in plant-based meats as well as in like normal meats. But I read that there was, I guess, more of an absence of saturated fat, I think, that I feel like cultivated meat could provide, but plant-based meat couldn't. I don't know if is that true?
Well, so certainly with the like plant-based meat, you do need to like supplement it with some types of like fats. And I think at least, well, I went to a talk for like Impossible Meat, so that's why I like seem to know a little bit more about it. But they like add a lot of like sunflower oil to their product to make it like like when you like fry their patties, they kind of ooze with oil. So I think that's like the sunflower oil. So certainly you need to like, that's one drawback is that that's such a complete reductionist like approach. Like you have to combine everything together. And for the like cellular agriculture technologies, you also need to like culture fat cells, or like they're called adipocytes, and mix them with the muscle cells. And also, as Laura mentioned, the connective tissue. And I think another drawback that I was thinking of was that some plant products have like an unpleasant like beanie like smell or taste. Like so that's like something that all these companies are like working on like reducing. And the question is like, should you modify like some of your organisms to reduce that kind of off-putting taste slash smell, or should you like have some additives that improve the taste? So like that's gonna cause a lot of, and this is all just to make it taste more like meat. Like I think in general it's not like a disgusting smell, it's just like not the smell that you expect when you eat meat. Okay, any other comments here?
I'd just like to add a comment that from a nutritional point of view, in general, it's good to greatly reduce saturated fat. So it's an advantage. Most people think in plant-based meats that there is very little saturated fat. And I think beans are delicious and smell great.
I do too, but I think even the woman, I think she was the vice president who gave the talk from Impossible Foods, brought that up, that it's a challenge that it's not with the consumer who's going in there to buy meat. They're not expecting that beanie taste. But when I make rice and beans, I'm totally fine with it.
You're right. You're right.
Yeah, and to that end, with the one thing that I found really interesting with this, with the reductionist idea where you get to add all the components together, is you really do have control over some of these, some of the nutritional drawbacks. Like they base for Impossible Meat as well as Beyond Meat, you have essentially the same product as the burger that you buy in the meat section, but they don't add any cholesterol, obviously, because like why would they add back cholesterol to it? So I thought also from like an allergy point of view as well, that's really awesome. And the idea that we might in the future be able to completely personalize our food and still end up with a very similar end product. Although that's not a drawback or a weakness, that's a benefit. So that's okay, we can talk about both. Okay, how about we move on to the next question? Okay, so this one I expect some of you guys will have a lot of comments on. This is from a talk by the same woman who wrote the first article. It was only like 14 minutes long, and it was really digestible. So we were inspired after listening to her talk to ask questions of like what would make these like cellular agriculture products natural, and how is it similar or different to GMOs, as well as like what will be the most difficult to convince the public to understand this and also eat this. And along this end, I hope that you guys will entertain me. I made a small poll, so I just want you guys to like answer these 4 questions. They're just yes or no questions, and I basically want to understand like, first of all, would you guys eat cultured meat? And this is specifically lab grown meat, not like not the Beyond Burger, the Impossible Burger. And then I would put it into like hierarchies, like, do you think you could convince your sibling? Do you think you can convince your parents? And then do you think you could convince your grandparent? to eat this. And in my head, I expected like most of us would be able to eat or would maybe want to try the cultured meat. And then maybe not. There are some reasons why we wouldn't, and we can talk about that. And I expected to get like lower and lower as we get out to like some of our like older relatives, which seems to be the case. I think you guys all voted, so I'm gonna share the results with you. So I think the majority of us would eat cultured meat, and we would all apparently be able to convince our siblings to eat cultured meat. But it reduces with like your mom or your dad. And I think most people don't think they could convince their grandparents. And I'm really curious to hear if the person who said no to eating cultured meat is willing to share. If not, that's fine. Last time we had somebody who said no because they actually have an allergy to meat product or something that's in meat. So that's why they wouldn't eat it. But if you guys feel comfortable, please tell me like why you don't think like your mom or your grandparent will eat this.
I'm the person that wouldn't eat it, so I'll start with that. And it's two things. One is I'm vegan, so I'm wouldn't eat anything that has the not donated plug of meat that they start with to culture it. That's not a donation. That's taking a piece of somebody's body to do something with it. And there's also the health issues. There's eating plant-based proteins is a lot healthier than meat-based proteins. And I would be worried about, I would just be reintroducing the reasons that cancer and heart disease and all that. So I'm worried about it from a health point of view.
That's really interesting, actually. I was just talking to my mom yesterday about this, and she mentioned the same thing. And it's like, that's something that didn't even cross my mind as I've been researching this. And she's very pro like plant protein in general, and really hopes that I go into like a plant-based company. But I did see your comment on the Slack channel, Linda, about, or maybe you mentioned it during our talk, that this is not like a donation. And it's actually something that I talked with Lauren about yesterday, that we're actually not even sure at this point in time whether or not people collect these biopsies, like if they actually use anesthesia, number one. And number two is it might even be the case that they just go to the slaughterhouse on the day that they're killing the cow and take a little bit of product, which might just be because of limitations of like what they're allowed to take. Like in my head, I can't picture a farmer right now who would be like, sure, you can administer the anesthesia to my cow and like take this little piece of muscle tissue from it. So I also agree that it's not like at all probably comfortable for the cow to donate this.
Well, I mean, just think about what if you actually do have this in a production situation, you wind up having cows in factory farms whose job it is to live their life as long as they can, having plugs of flesh taken out of them.
Yeah,
it's not like, and they're not going to use anesthesia. I mean, think about what they do right now with the tail docking and the debeaking and all that. They're not going to use anesthesia if they could possibly get away without it.
Yeah, I think that's an overly optimistic like view of how that biopsy works. But if it helps, you probably know this as well. But what we're trying to do is like find the perfect like cow cell line and then like keep that like frozen. And then constantly just go back to that stock of the frozen cell and expand it. And that would be, I think, like the optimal, if we have just like a couple different cell lines. And I think Lauren yesterday when we were debating about this pointed out that the only problem there is that we're limited to like the cuts and like the tastes that come from each of those cuts. Like if we just have one cell line, we'll all be eating like the same meat for the rest of time. And like that will be the future of food. And
that would be awesome, like Henrietta Lacks.
Yeah, that's exactly what I mentioned to her. I was like, that the only thing is that obviously that cow didn't know that it was like gonna be the future of science, just like Henrietta Lacks. And for those of you that don't know, like Henrietta Lacks was a woman who had like cervical cancer. I forget when, like in the like, was it 40s or 50s or even before then? And she she was also a Black woman. So at the time they, without her consent, took cells from her tumor and they were just able to culture them as like, it was one of the first human cell lines they were able to culture in vitro, and nobody ever told her about that. And also they like have a whole bunch of medical developments from those cell lines, like curing or finding the vaccine for polio, for example. And her family still to this day hasn't received like any financial compensation, and they still all live in Baltimore. This happened at Johns Hopkins, and you know, it's just a kind of terrible situation that happened. Although her cells have been instrumental to all of us, basically. So that's like what would happen with this cow, basically, is if we found the perfect cell line that was delicious and worked really well for all these applications. It would be great in that we never had to kill another cow, but kind of still unfair to the original cow. Okay. Any other comments here?
I think if you guys have watched the commercial for Eat Just Chicken, the chicken nugget, Good Meat, I think they screened it as while they're eating the chicken nugget, the chicken is right beside. So it's like the chicken is still alive while you're eating the chicken meat. And I think that it's, I'm not sure how, like probably is for commercial purposes or whatnot, but yeah, I feel like that's where the cellular agriculture companies are going toward. And I personally have had the cell culture chicken nugget actually, and I don't feel like it tastes like chicken, but it's pretty convincing.
I think that's kind of, I find that even weirder, like looking at the thing that you're eating, but
I know.
maybe
I
some
totally
people do
agree
feel
with that.
better when they look
Yeah.
at that.
Mhm.
Whitney, you got to try the actual cultured meat, is that what you said?
Yeah, I can send you pictures afterward. They made it into chicken nugget because it's less controversial that way, because chicken nugget, to be honest, I'm not sure how you can compare that with real chicken, because it's a mishmash of all things not chicken. And they also added plant-based protein into the nugget, so it's not 100% purely chicken. So yeah, that's controversial also. And they fried it, and it's like Impossible Burger if you grilled it on like that with such charcoal taste. It covers all the artificial taste, so it's hard to tell how approximate it is with real meat. But at the same time, yeah, I understand the topic is very controversial. It's like, would you consider this product as vegan, and would you consider it as GMO or not? It is
Well, I mean, I think I think we're all in disagreement about whether or not it is vegan, according to what like Linda brought up. But yeah, and like Lauren was mentioning that it's preferable to like factory farming, this taking biopsies. But I also feel like what could end up happening if we don't find the perfect cell line and freeze it is what Linda mentioned, where we just switch to like a factory farming just to like take bits and pieces of cows. Although hopefully we wouldn't need as many in that case, but it's definitely going to be interesting to see what the future holds. And we're over time. And I just want to mention that the last question that we had was about this like document that we found that is like 90 reasons to switch, or not switch, 90 reasons to consider like cellular agriculture. And I just want to tell you guys, it's I found the article, it's just literally 90 reasons, and it's a very pretty, like it's very digestible format. So if you have anyone in your life that's skeptical, or you yourself want to see lots of more reasons to consider this, I just highly recommend downloading that. But I think to respect your guys' time, we can like wrap up here. And our emails are on the last slide here. And I also want to mention that to Linda, I'm sorry that we don't know much about the TMAO, but if you don't mind putting some documents in the Slack channel about it, I'd be happy to read them and like talk with you separately about it. Just because I want to know more about it too.
Super, thank you so much. And I want to end just saying, I'm saying that I'm after reducing the suffering. So I'm so thrilled that y'all are working on the growing meat from cells, and that reducing the number of animals involved is a good thing. So even though I wouldn't eat it, I think it's a good thing. And that's why I could convince my friends to do it.
Yeah.
So I don't want you all to go thinking I'm saying it's a terrible thing.
Yeah, thank you. I didn't think you thought it was terrible, but I also agree. Like I honestly don't care what anyone eats in terms of like their health, as long as it's not affecting the environment and like affecting me in that sense. So like even if these are like animal proteins and they're more harmful for you, as long as we're not having so many like of these like fields and like deforestation, like that's also a huge benefit. Okay, thank you guys all for coming. And these are our emails. And we mentioned last week or yesterday that you guys could also add us on LinkedIn if you want. I think you can find us through these emails.
Yeah, you can just type in Lauren Blake Hopkins, Molly Gordon Hopkins will probably show up. Yeah, and feel free if you have any further questions, and we can continue the discussion in the Slack too. If you guys are down for that.